This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 37, recorded on July 12th, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
How are you?
How you doing, Elio? How's San Diego?
I'm considering small things. San Diego's fine, sure.
Yes, always. And you're fine, and you've gotten over ASMGM, right?
Yeah, I have.
Good. Also joining us today from Medical University of South Carolina, Michael Schmidt.
Hello, Vincent and Elio, and we have Joe with us today, too.
Yeah, returning after a long absence, as they say. From Yale University, Joe Handelsman. Welcome back.
Well, thank you. It's great to be back.
Yeah, we like having you on, and so we're going to have you 3 episodes in a row if everything works out well.
Oh, lucky us, lucky us.
Summer is so special. I love it. I can do things like this.
Unless you're called to the White House, I suppose.
Oh, well, let's hope that doesn't happen this summer.
I wanted to ask you, on your Skype logo, there's a little glowing image here. I can hardly see it. Do you know what that is?
No.
It looks like a bacterium with some green fluorescence on it. You don't know what someone must have put it there for you.
Yeah, I'll have to go look. That's very interesting.
Yeah, on Skype you can have a little image of yourself, and yours is this GFP-looking thing. I just wonder.
Well, we do have some GFP bacillus, so that must be it.
That must be it. You also have Madison, U.S. still on your Skype, but you're not there anymore. We know.
Oh no, I should change it. Thanks for letting me know. See, I don't call myself on Skype that often.
Yeah, that's right. I know. I don't know what mine looks like either. Anyway, it's great to have you back, and today we actually have a theme, amazingly. 2 papers that we have are related, and I think the title of this show will be Conflict and Cooperation.
Hey,
what do you think of that? Is that good?
Yes.
That's good.
I actually stole it from one of the papers, but it makes sense because we have 2 papers about switching between mutualism and antagonism or pathogenic states. And the first one is a paper that Elio brought to my attention in a journal that I'm not sure I have ever looked at, the Journal of the American Chemical Society, or at least not for a long time.
It's good for the soul.
You think? And there's a lot of chemistry in this paper.
But actually, there's another paper that deals with this. It's and this is published in Nature Chemistry.
Mm-hmm.
And it's by the same author, so it's a similar paper.
Does it predate this one, or?
Yeah, it predates this some.
Yeah.
And it has a more fun title.
That's right. The Jekyll and Hyde Chemistry of Theobacter Galaissiensis.
That's a good title.
I'll explain that.
That could be a good title also, Jekyll and Hyde in the prokaryotic world,
bad.
Something like that.
Yeah, I like that. Let's use that one.
In the microbes. Call it microbes because it's both.
So microbial Jekyll and Hyde, something like that.
Yeah.
Good.
Hey, you're on.
The title of the other one is, it's less good, Rosiobacticides, small molecule modulators of an algal bacterial symbiosis. I like this one, Elio, because it brings back our friend Emiliania.
That's right.
Which we talked about
We recently talked about this amazing little alga, a couple of microns across, which can be one of the biggest inhabitants of the oceans. It causes blooms which consist of huge areas. I mean, thousands of, several hundreds of square miles or thousands of square miles. And it can be seen from outer space. The reason being that these guys are covered with little beautiful plates of calcium carbonate that have sort of like an armor of these plates. And the calcium carbonate is white and reflects the light. And so you can see these blooms very easily from satellites. Now, what we talked about the other day is that when these blooms collapse, they collapse apparently because viruses attack them.
Mm-hmm.
We have a real nice theme there. And it turns out that there's other possibilities and that the situation is a little bit more complex. And it involves bacteria. And so we're going to talk today about a duo of these Emiliana Huxley or EHUX, as it's known. It's a coccolithophore, which means a stony ball. And a certain bacteria called, from a group called Roseobacter, of which this guy Theobacter is one. Let me make a small digression about Roseobacters because they were not known until 1991. They turn out to be among the major constituents of the bacterial flora of the oceans. They can make up as much as 10%, 20% of the total bacterial biomass in the ocean. They're a large group and it's a heterogeneous group. They all are alpha proteobacteria for those who care about such things. And they're very varied. It's a large heterogeneous group. But what they are known to do, and we'll come to this in some detail later, is that they take a compound which is made by the algae, namely dimethyl sulfonylpropionate, which I'll call DMSP from now on. And DMSP is made by the algae apparently as a cryoprotectant. The algae find themselves into very cold waters as well as warm waters. And also it works apparently as a osmoprotectant as well, being in the ocean of high salts. So this DMS stuff is soluble, and when the algae break up, they put out huge amounts of this. The amounts are incredible. They're 10 to the 9th tons per year. This is not a small part, and it's part, of course, both the carbon and the sulfur cycle. 10% of the weight of the algae is the MSP. Okay, that's good enough. But what happens is that the MSP is converted into DMS, dimethyl sulfide, by these Roseobacteria. And here is a story that takes us in a completely unexpected direction, maybe. I think, unless we mentioned it before. And it's the following, that DMS is volatile. So it goes up into the atmosphere. It is converted by sunlight, by ultraviolet light, into other sulfur-containing compounds, which become nucleation centers and make water droplets. In other words, they make clouds. So these dying algae give off the MSP. The MSP becomes DMS, goes up in the clouds, and it alters not just climate, which is the long-term phenomenon, but they alter the weather, which is what happens in the next half an hour. The importance of this is not quite clear because there's lots of human-produced sulfur that goes up in the air, but this is important. So we have a connection between photosynthesis in the oceans and climate and weather.
So we had mentioned this cloud issue last time, but I don't think we discussed that a bacterium actually converts the DMSP. Right. So that's something new for me.
Right. And they do it with gusto, and they use it. They do it two ways, actually. The DMSP becomes DMS, and by another pathway, it becomes methane thiol, which binds to metals and which is also a direct source of carbon and sulfur for the bacteria. So this is foodstuff for the bacteria in part, and in part it's something they convert. So the Roseobacteria matter. They go along with EHUX. That is, when you have big blooms of EHUX, you have as much as 20% or more of the bacteria are going to be Roseobacter. When the blooms go down, there's less Roseobacter. There's a good correlation between them, both geographically and temporally.
So it's said that they're associated with the EHUX. What does that mean? Is that they're
that's a point which is not clear. They are thought to be symbionts, but the evidence is more this kind of circumstantial evidence I just mentioned, that they go along in time and space. There's a thought that they are symbiotic also because the EHUX provides a surface on which they can make biofilms. But I don't see the evidence for that yet. I mean, it sounds plausible, but it needs to be looked at, and I imagine it will be looked at, and maybe it has been already. I'm not aware of it, though.
The other thing to really appreciate is that the area that this can go on is measured in 10,000 to 100,000 square kilometers.
That's right. That's right.
I mean, that's just mind-blowing.
It's a bloom that's the size of Ireland.
Yeah. I mean, if you think about, climate change and the importance that I don't want to spoil what we're going to do, but it really drives home the importance of understanding how something like a small molecule can have almost a global effect.
Right.
When you consider La Nina and El Nino and cold and hot water, and actually what triggers the story that we're going to get to here.
Well, there's also the important, besides this business of the change in the weather, these algae become a sink for CO2 because they take CO2 and make calcium carbonate out of it, which is insoluble.
And it sinks.
The calcium carbonate, right? Yeah.
Well, it's literally a sink and a sink.
Yeah, because the shells go to the bottom, right?
Down and they deposit in such a huge way that they become the White Cliffs of Dover.
Yeah.
Made up of Emiliana Huxley shells. So if we were tampering with that, the amount of CO2 that would be free would be much higher, with the consequences that we predict of climate change. So we have to be eternally grateful to EHOX for sopping up a lot of CO2.
And that's why the pH of the ocean is so important as well, because it coordinates whether or not this calcium is going to come out of solution.
Mm-hmm.
I mean, go back into solution, I should say.
That's right.
And these are also the source of half of our oxygen on Earth as well. People always think that the oxygen comes from trees, but half of it comes from the ocean, and it's largely the micro or phytoplankton that make it.
Exactly. So these guys are about as crucial to our survival as anything can be, right? We can really be thankful to EHOX and the associated bacteria.
Mm-hmm.
Has anyone ever seen a EHOX bloom of us here on this show?
No.
Have any of you seen one? No.
But I've seen pictures from ships.
Yeah, you can see
It looks like the white seas. They call them the white seas.
Yeah.
You can see it from the deck of a ship that the water looks whitish.
That would be a cruise for microbiologists, a EHOX bloom finding cruise. You could charge people to do that, like whale watching. Instead, you take them out to see EHOX blooms.
Yeah, and you can scoop them up and look them under the microscope. They're so beautiful. You need an electron microscope on board, though, because to appreciate the beauty of these plates, you need that. You need a scanning scope. And by the way, I really recommend our listeners to go and Google image Emiliana Huxley or EHOX, and they'll see something startling. That's absolutely gorgeous.
The individual organisms are exquisite. I mean, there's been so much talk about the symmetry in biology, and I think this is one of the most used images of EHOX in looking at symmetry, because it has these exquisite plates all over the surface of the alga.
It's like a jewel, doesn't it?
Mm-hmm.
Yeah. Well, so here the story. The story is the following. Normally, when EHOX is happy, the associated bacteria apparently detect a signal from the EHOX, and the signal is weird. It has to do with, hold on to your seat, lignin. Lignin is something associated with trees, right? Well, it turns out there is lignin or lignin-like compounds in a whole lot of algae: red algae, brown algae, green algae. Okay, and in the group that EHOX belongs to, which are the Haptophytae, has not been known to do that, but they have compounds like it. And lignin, as you know, are very messy aromatic compounds that nobody, I guess, wants to study. Everybody wants to get rid of lignin because that's the way you could convert trees into ethanol or something. But anyhow, some of the compounds derived from lignin or from lignin metabolism stimulate the bacteria. And by the way, DMS P itself is a chemoattractant.
Hmm.
So there's something that the algae needs from the bacteria, and it makes sure the bacteria are attracted by flagellum motion, by regular chemotaxis to them. But these compounds are signal molecules, and they set off the Theobacter to make two classes of compounds. One are antibiotics. I never heard of these. These are a thing called tropodiethylatic acid, whatever it is. It's an aromatic compound, and it has bactericidal activity against a number of bacteria. And the thought is maybe it just keeps the EHOCs from getting infected, invaded by other bacteria. Nobody knows that, but it's a good thing to have, apparently. And the other one is even more startling, are auxin-like compounds. In other words, growth promoters for the algae, like phenylacetic acid, which are known to be such auxins. And these are made by bacteria, so the bacteria are warding off possible invaders and stimulating the growth of EHOCs. And what do they get in return, the bacteria? Well, as we said, they get nutrients. They get the DMSP, which they like, certainly, or methane thiol. And possibly they get this way of a surface on which they can stick. I think we are all now brainwashed to believe that biofilms are probably the way of life for the overwhelming number of bacteria in the environment, and so sticking is important. Anyhow, that's the normal situation, and everybody's happy. However, when EHOCs decays, and there is no mention in these papers, it seems to me that the decay could be initiated by the viruses that we talked about.
Mm-hmm.
There's no mention of this, but this is apparently correlated with the collapse of these blooms. So let's pretend that the bloom collapses because of the viruses, and now the algae put out other compounds, and these other compounds are also aromatic compounds, and they are effectors of a physiological, biochemical activity of the bacteria. Namely, they make anti-algal compounds. The effectors are things like p-cumaric acid. You can look it up if you want to. There are others which are even more effective, which apparently they didn't study very much. They have names which I never heard of. I'll give you some. Ferulic acid and synaptic, not synaptic, but synaptic. Anyhow, these are compounds that at micromolar concentration make the bacteria make these compounds. And these compounds are, let's not get confused, they're all aromatic compounds. Everything that we talked about so far, the antibiotics made by the bacteria, the auxins made by the bacteria, and now the effector made by the algae is p-cumaric acid, which is an aromatic compound, all rather related. And finally, these anti-algal compounds, which are called What are they called?
Roseobactin.
Roseobactin.
Or bacticides.
Roseobacticides. And there's a whole collection of them. And by the way, and so they affect other algae. So what do? Well, nobody really knows, but the idea is maybe they just kill off the bloom, which is half dead, and just get rid of it. Or they impede other algae from occupying the site that EHOCs would like to occupy. I must say, the ecology of all this is still up in the air.
The organic chemistry. If you're really interested in this sort of thing, many of us took organic chemistry as undergraduates, and those of us who took graduate level organic chemistry, when we were studying for qualifying exams, we're terrified about synthetic reactions. If you look at some of the chemistry associated with these compounds, there is no way on God's green earth any human could ever make these compounds in a flask. This really drives home the wonder of microbial metabolism and how they're able to carry out these very sophisticated chemical reactions in which you have disulfide bonds that are effectively making a 4-member ring off of an aromatic structure. I mean, some of these things are just mind-blowing on how they were able to take these oxygens and sulfurs and chirally rearrange them so you get these different activities. And, we're talking about the need for new antibiotics constantly, and here we have something so elegant showing us antimicrobial activity in something that obviously has been selected for a mutualistic opportunity for these creatures.
To emphasize that, compounds have no relation to any compounds, natural or synthetic.
Yeah.
A class of their own. So we've got new chemistry here. So what happened is, in order to determine the structure, these guys went to town, and it pays to look at least at this article in the Journal of the American Chemical Society to see the great length. If you look at the supplemental material, it's just spectral data upon spectral data upon spectral data, and it turns out this is hard stuff. If I read between the lines, I'm not as versed in organic chemistry as some of you, especially Michael, but they start out with X-ray crystallography, and that sounds good, and that doesn't tell them enough. So they go into a whole collection of mass spec and just go on and on and on, and MRs that I never heard of.
With energy levels, I mean, the NMR energy levels, it would pay collectively all our utility bills for a year. Yeah, I mean, NMRs are not cheap to run.
Well, there's HRMS, over MS, and so forth. It just goes on and on and on. So it took some very clever chemists, and by the way, these clever chemists, some of them are, one of them at least is an author of the next paper, John Clardy. So we're going to hear about this again. Anyhow, their organic chemistry must, I assume, be superb, because this is not easy stuff. This is not your ordinary little small molecular weight compound that you can just figure out the structure of. And by the way, there's a whole panoply of each compound I mentioned is actually represents a class of compounds. The antibiotics, the anti-algal, the effectors. So this is a zoo. And Michael, I'm glad you made this point that this is just the richness of the microbial world in chemical terms is just amazing.
Well, this is really important. And again, this goes back to some of the emerging trends at the general meeting, where we saw the introduction of MALDI mass spectroscopy as a common tool in the diagnostic laboratory. And what MALDI mass spec does is it looks at relatively low molecular weight protein complexes that are principally soluble inside bacterial cells, and that's effectively how they're going to go off and identify new bacteria. And so it's sort of good that we're doing this to tell our friends in microbiology that you got to occasionally go over to the dark side of the American Chemical Society and look at their papers, because you're going to have to be able to understand some of this chemistry, because it's going to creep into the Journal of Bacteriology, it's going to creep into Applied and Environmental. This chemistry is not just going to be relegated to ACS. It's actually going to become a component of our discipline by virtue of the fact of the power of these tools.
Actually, can I add to that something that we ought to discuss sometime? This is a digression, but it's on this theme. There's a guy named Peter Dorenstein in the pharmacy school at UCSD. You know his work, do you?
Yes, doing very similar work with a chemist here. Amazing stuff.
It's amazing. What they do is they bombard colonies of bacteria with various bullets. I don't know what all they are. Some are iron, some are, I've forgotten what all.
Mostly mass spec technology.
That's right. It's all mass spec technology. And they collect what bounces off the edge of a colony or the middle of a colony, and out of it come series of compounds from ordinary bacterial colonies, which the world has never seen. And so, who knew? This is absolutely incredible that there is compounds in our ordinary. By the way, E. coli of all things is the dullest of all the bacteria they tested. It has the fewest of these compounds, and maybe it's because they tried laboratory strains. I bet you that's what it is. But you have experience with this, do you, Joe?
Yeah, I think mass spec and looking at the whole profile of small molecules that bacteria make is going to be sort of the next level of systems level biology. We've looked at the genome, we've looked at the transcriptome, we've looked at the proteome, and I think the metabolome, but not of central metabolism, of these more of these secondary metabolites that aren't required for basic growth but are required for other things, are going to be the core of a lot of the future of microbiology, largely because these are the molecules that are used to communicate with the environment. And that may be the abiotic environment, or it may be organisms, as we're reading in these papers. And the ability to detect those compounds has always been the limitation. The complexity of the chemicals, the difficulty in purifying them are just staggering. And so the mass spec capability of today and the power of these machines lets us have more of a total portrait, sort of like getting an entire genome. We can see the entire metabolome, and it's really quite marvelous to see, one, how complex it is and how many different just beautiful molecules bacteria make that we never knew about because we couldn't detect them before, but also how they're modulated by other bacteria and other organisms in the environment. And I think it's just a network of chemistry that modulates the activity of individuals as well as communities. I think this is going to be the core of community biology.
And it's worth one more digression about understanding how to culture bacteria, because as Elio pointed out, they're doing this often off of colonies, and a colony is really a population of cells that's in maximum stationary phase, if you will. Right. And being able to grow them on agar plates, and I'm not talking LB, I'm talking, think about growing them on a defined medium in which you can put a heavy amino acid using say, for instance, heavy sulfur or heavy nitrogen or heavy carbon, so that you could then track it. And so that way you can compare and contrast.
Physiology. You can do physiology.
Yeah, old-time physiology. So enough of these digressions.
I've been preaching that people who are going to microbiology should study physics. Turns out they should study chemistry also.
Yeah. Well, it's really the physics of chemistry, because it's time of flight, and you really have to understand how these ions fly through the air with the greatest of ease, as they say.
Elio, can I ask you a question? So in the ACS paper, they make the statement. So when the bloom is falling apart, the EHUX bloom, it causes these bacteria to produce compounds that kill the EHUX, and they say it's to secure the food supply provided by the dying host. It sounds like they know this, but is this the case, or is that a speculation?
I'll tell you, like always, when you have a fantastic new phenomenon that you study, you can't do everything at once, and so usually the thing that remains for last is the ecological meaning, the ecological studies. And I would say that, and it's because they're more difficult. I mean, it's easier enough to do the rest by comparison. To do ecological studies means you have to have a technology that you can adapt to working in what they call a microcosm, a liter of water from the ocean or something like that. Yeah. So I think very little has been done along those lines. I think the speculations are smart, are certainly plausible, but I think we need quite a bit more work, and I bet you it's going to come. I don't doubt it. Could I sort of sum up what this paper say? Because by now we've wandered all over the place. Okay. So the proposal is the following, that when all is said and done, EHUX and Rosebacter have a close relationship. Rosebacter acts in one of two ways. It can be a mutualistic organism which benefits from the presence of its partner EHUX and makes EHUX gain from its activities. And that's sort of the steady state of normal growth, if you wish. Then when something happens to EHUX, the bacteria become a little bit ferocious, and they apparently kill whatever is left, so they'll get rid of that, and they become parasitic. And the mechanism of action of this is all chemicals which are related by virtue of being all coming from phenylalanine, essentially. And so they are all aromatic compounds which undergo a series of very complex chemical reactions, ending up in one of these two states. And just precisely what sets off the change from one state to the other is, I think, yet to be determined. But it's there. I mean, the phenomena are clear. How it works and what sets it off needs a little bit more work.
Yeah, figure one is a good summary of all of that. Yeah. It's very sad, actually. So when the EHUCs get old, the bacteria don't want them anymore, so it kills them and replaces them with younger species.
Don't you remind me.
Sends them to the bottom of the sea, literally.
That's where some of us are doomed to end up.
Wow. This is a great story. All right, so you're done with that, Elio? You want to move on?
Yes, indeed.
So I wanted to just point out one other little quirky item in this paper. So Elio had mentioned that the DMS produced by the bacteria induce nucleation of clouds, and presumably that's nucleation of raindrops, which often start out as ice drops, actually, tiny little snowflakes. And then as they come down to Earth as rain, they melt, and that's why we see them as rain. This is the second example of bacteria that may play a major role in weather in the clouds. We've talked about the importance of these bacteria in the ocean, but there are other bacteria called ice-nucleating bacteria that are known for their activity to cause supercooled water, so water that's below the normal freezing point, which is 0 degrees, to freeze. And so water can supercool below 0, and if it doesn't have an ice nucleus, then it will just keep cooling and cooling. But with the appropriate ice nucleus, it will then immediately freeze, right? Very, very quickly, right in front of your eyes. And one of the best ice nuclei known are these bacteria, the ice-nucleating bacteria, and they have a particular protein that causes the ice to form. One of the things that's been found is that these bacteria, although they live typically on the surfaces of plants, will leave the surfaces of plants, lift off on sunny, hot days, and perhaps, and this is still speculative, but there's a little bit of data behind it, go up to the clouds. And perhaps they're playing a role there in stimulating ice formation that becomes rain to bring themselves back down to Earth. So I think this is going to be one of the real surprises, because we think about bacteria as doing a lot of things, good and bad, but I don't think most of us think of them as regulating the weather. And so I think this is going to be a really interesting field of microbiology in the future.
I'm sure glad you brought it up. That's really a great story.
It's almost as if they're the sprinkler timer on the plant.
Exactly.
Because when are you supposed to water? You're supposed to water early in the morning or late at night. You're not supposed to water in the middle of the sunny day. And so the bacteria go up to the clouds, and eventually it'll take mass action to cause the rain to come down just when the plant needs it.
So maybe this is like the equivalent of plants doing a rain dance?
Movement. We're anthropomorphizing everything today.
Yeah, we're anthropomorphic and sentimental, and we're funny.
Isn't there some use of these ice-nucleating bacteria to protect crops in some way?
There are two uses. One is to make snow. So they're often used on ski slopes.
That's right.
To help make snow because it requires less energy when the ice-nucleating bacteria are available, because you can actually get the formation of the ice crystals at higher temperatures, so you don't need as much energy to cool the water. But the big controversy that I bet is what you're remembering was the ice minus bacteria. And what people in California did was to delete the gene that encodes this ice-nucleating protein from the surface of from Pseudomonas, and which is one of the ice-nucleating organisms, and then use that to protect crops. And the idea was that the ice minus, the one without the ability to induce ice, would be competitive with the ice plus bacteria, and by reducing the populations would reduce the probability of freezing at slightly sub-zero temperatures. Right. And it worked. But interestingly, after a few weeks, the Ice Plus bacteria took over again, even in the absence of cold weather. So there's some advantage to being Ice Plus.
Well, it's probably the sprinkler effect. The plant's probably the plant's probably giving the Pseudomonas with the Ice Plus something that enables it to go off. There's this tour de force. Since we're keeping with our theme of mutualism, it's interesting now as you begin to think about this as we move to this next paper about pathogens and mutualistic relationships between bacteria and insects. So I think it'll be a neat transition.
Mm-hmm.
All right, our next paper was in Science last, wait a minute, is it just published this month?
Yeah, July 6th.
July 6th, 2012. Single promoter inversion switches photorhabdus between pathogenic and mutualistic states. And I thought you would like this, Joe.
Oh, I love this. Yes. I pulled this off the web even before my copy of Science appeared on my desk. I thought it was one of the more interesting and biologically rich papers I've seen in a while. But our discussion of the other paper and of the role of chemistry sort of brought to mind an issue that I think plagues a lot of biologists, and that is that chemistry is becoming more and more important, I think, in most fields of biology, but particularly in microbiology, because the small molecules are how bacteria communicate, and we have these two wonderful papers demonstrating that today. But the irony and really challenging aspect of education for many biologists today is the fact that the organic chemists are not training the right kind for us to collaborate. And so John Clardy, who is the person we mentioned as being common to both these papers, is one of these tremendous natural products chemists who can take, seems like any molecule and figure out its structure. He's one of the early people who used X-ray crystallography of proteins with their small molecules bound to actually determine the structures of some small molecules. He's just a master at this. But unfortunately, because the drug companies, the pharmaceutical industry has stopped using natural products as much as they did 20, 30, 40 years ago, there's less of a need to train this kind of chemist. And so the result is that the pharmaceutical industry drives a lot of the fields that the chemists get trained in. And so the result is that for those of us who need these chemists to collaborate with, they're getting to be in short supply. And so I think there's just a major challenge for microbiology. Maybe we should create some fellowships to train chemists in natural product chemistry or something. I don't know. But I think these two papers show us the power of these small molecules and of the chemists who have the talents to work with them. So this paper is a single promoter inversion, switches photorhabdus between pathogenic and mutualistic states. So I wanted to start by just unpacking the title. And the first part that's important is this organism photorhabdus, which lives in two different conditions, probably others, but the two important ones are that it can form a mutualism or mutually beneficial relationship with nematodes. And the nematodes, in fact, can't go through their full cycle of development, moving from juvenile to adults, without the bacteria present. So there's a real dependency there. The bacteria also get a nice home inside of the nematode, but the nematode offers them another advantage, and that's that the nematodes can infect insects, delivering the bacteria to those insects. And when the bacteria get out of the nematode and into the blood system or the hemolymph of the insects, they can kill the insects. So that's the pathogenic state. So that's why we're talking about a switch between mutualistic and pathogenic states. And so it's a mutualist with these little worms, the nematodes, and then together with the nematode, the bacterium serves as a pathogen for the insects.
But it never, in this pathogenic form, it never hurts the nematode, right?
No, that's right. It doesn't.
And the other thing about the pathogenic state that Joe's probably going to get to in a minute, but I will introduce this because I'm in Charleston, and one of the happy circumstances is we always have these Civil War buffs. So I was asked once by one of these Civil War buffs about something called angel's glow. And there was reports during the Civil War, or as it's referred to down here, the War of Northern Aggression, that soldiers' wounds used to glow. And it's now been worked out that the microbe that's responsible for these wounds glowing was a consequence of this particular bacterium, Photorhabdus luminescens, that were making antibiotics that were actually facilitating the cleanup of the soldier's wound. And obviously they got the wound, they fell down into the dirt, and what's in dirt but the nematodes? The nematodes came in, dropped the bacterium, and it's defecated out into the wound area, and here this bacterium is actually delivering an antibiotic to preserve the soldier's limb.
So the worm thought it was an insect and deposited these bacteria.
Yeah.
Wow.
By the way, this even happened in the First World War in the trenches in Europe, where, again, the idea was that if you could see a glowing light on a wound, you were better off. You're on the way to recovery.
Wow.
It's incredible.
It's a great story.
Yeah. And they are beautiful bacteria. They're so luminescent. I mean, as the story tells, you can see them just with a naked eye. In a dark night, you can see the wounds glow. So they're pretty remarkable organisms. And just this switch from the pathogenic to mutualistic state in itself is so amazing, because they have such grossly different effects on two different hosts. I mean, opposite effects. In one case facilitating life, and in the other one killing the organism. And so what this group found is that there's a very dramatic switch, and it's at a very particular point in the genome that they found the cause of the switch from the pathogenic to mutualistic state. And so it's not just a simple adaptation. People had speculated many different mechanisms for how the bacteria might begin to move toward the pathogenic state. And in fact, it's not a gradual shift. It's not multiple genes. It's one inversion of a promoter that causes the switch. And so this promoter is the feature in DNA that drives gene expression through transcription. And when it's in one position, it drives the pathogenic associated genes, and in the other, it drives the mutualistic genes. And so it's called the MAD switch.
It's great.
And I love the MAD switch. And the main things that it drives are the production of fimbriae, which are these sticky attachment organs that the bacteria have. And
also known as pili, right?
Also known as pili.
Yep. And those allow them to stick to the intestine of the nematode. And so that's a really important element, and the first thing that gets transcribed from this promoter. And so there are a few examples of this in microbiology, but none that I know of that are just as clean and elegant as this one, where an invertase, an enzyme that clips out the DNA of this MAD-R site and flips it, makes it the switch that goes one way or the other, changes it from a very good bacterium for one host to a pathogen of the other. And there's a whole fleet of changes that come along with that.
Before you do that, before you go into it, could you remind us what's the difference or relationship between this phenomenon and phase variation? That is a well-known, well-established thing that Michael pointed out last time we talked about it.
Well, in Salmonella, it is very similar to this. It's a little tiny switch, and it's the repressor that is absent that causes the flop of it. So it's a small swap, just like this, and it's a transposase instead of an invertase, but it effectively does the same thing. It's not that big of a chunk of DNA in the Salmonella typhimurium type of phase variation. Coli may have a little bit something different, but it's very reminiscent of that. And I found it absolutely fascinating how something so small, but you're probably going to get to this, but don't want to lose it, is that this mutualistic form of the bacterium is so much smaller in size than the pathogenic form. It's 1/7 in volume. So if you think about the volume of a bacterial cell, in this case, because they're in the Enterobacteriaceae, we can use the coli number of 10 to the minus 15 liters. So 1/7 in size means their volume drops to 1.4 times 10 to the minus 16 liters, which is even smaller in terms of thinking about the chemistry that has to go on, because bacteria, for better or worse, are all solution-based chemistry, and the number of molecules that can actually fit in 10 to the minus 16 liters.
Oh, but wait. You're surmising. You're starting out with an assumption that this is the size of E. coli. I think it's actually quite a bit bigger in the virulent form.
Oh, you mean this pathogenic form?
Yeah, in the pathogenic form.
Oh, it's in there.
Well, there's a picture with the 5-micron scale, and it's hard to tell in a fluorescent photograph because they glow over a larger area than they actually take up in terms of real space. So I'm not sure.
The M forms are 1.2 microns, and the P forms are 4.4 microns. In length, the diameter is 0.8 microns and 1.2 microns.
Okay, so Mike has a point. I'm sorry.
Mm-hmm.
Well, anyway.
So one of the amazing aspects of this switch is one that happens apparently randomly, and it's about 1 in 1,000 cells will switch from the P or pathogenic form to the M or mutualistic form. And when they switch, they acquire all these other characteristics. And as Michael said, one of the things they become are small cells. The other is that they're enabled to persist in the intestine, and that's presumably because they're expressing the fimbriae that allow them to stick. They also produce no crystal toxin, which is the toxin that actually kills the insect, and so that's not needed when they're in their mutualistic state, so it's not expressed anymore. They also lack the ability to produce stilbene and a whole bunch of these other small molecules. And this is the connection with the previous paper, that small molecules seem to be one of the key elements of these stories in terms of communication. And then they're also much more resistant to ciprofloxacin, the antibiotic. And so this switch from pathogenic to mutualistic may have some very interesting implications in other organisms, because there are other examples of small persister cells in organisms like Staphylococcus and others. And in some cases, it's associated with resistance to antibiotics. So this may have all sorts of implications in medicine, as well as in this kind of esoteric symbiosis out in the real world.
That's a good point.
So one of the approaches that they used in this paper that was a really interesting way to phrase it, but also to do the experiment, was they deleted the invertase, which is the enzyme involved in switching orientations of the promoter. And that made the organism lock, as they called it, in one form or the other. So they had one locked in the pathogenic form, and then the other one locked in the mutualistic form, so that they could study those forms without the this random variation of the mutualist one going at a rate of about 1 in 1,000 to the pathogenic, and the pathogenic one going to being mutualist at about 4 in 100,000 cells. So they didn't have to worry about that switching once they locked them by taking out the switch. And then they studied the characteristics of each of them, and they were that long list of things that I mentioned, as well as quite a few others that were quite different. And the difference was greater when they locked the characteristics by taking out the switch than in the wild type normal one that could go back and forth. And that makes some sense. If the entire population is locked in either the mutualistic or the pathogenic state, then you would expect the phenotypes to be stronger. And so they have really gotten to what I think is the core of what's always been fascinating about the Photorhabdus system, and that is an organism living with different hosts in these wildly different modalities. And it all seems to come down to this tiny little switch.
And this the key is that it's random, right?
Yeah.
It's a random switch.
Random when it happens. And so it happens at a predictable frequency, but there's no way of predicting which cell in a population will undergo the switch.
So basically what happens is when the nematode goes into an insect, it regurgitates these bacteria, and eventually the P form dominates in the insect because more of those are made, right?
Mm-hmm.
They kill the bacteria, and meanwhile, the M form is passed on through to the nematode's offspring by this adhesion mechanism.
But it holds on. It literally holds on for dear life, and it's just the luck of the draw, and it sticks. That where the persistent M forms stick is in that special area of the nematode that allows the mother to transfer it to the progeny.
And the nematode gets to eat the insect, I presume.
Oh, yeah. Because the crystalline, the other neat thing is that the crystalline, these crystals that are in there, everybody's probably seen Bacillus thuringiensis crystals that you can buy at, your Home Depot or your Lowe's or whatever, and, the natural form to sprinkle on plants that dissolves the peritrophic membrane of insects. But this is just another insecticide that's a natural form that the microbe is working in concert with the insect to effectively recycle insects into more insects, or more nematodes, I should say.
Is this used to control pests like the BT?
I don't think so.
Not yet.
Well,
Maybe.
I think there are some products, I don't know if they've actually hit the market, but they were under development. But as I understand it, the production of the nematodes is so expensive that it's been very hard to reduce to practice, because it's not just the bacterium, it's the cooperation. And that's, of course, the beauty of it, but it also makes it a much more difficult system to conquer at an industrial level.
By the way, it seems startling to me that the number of nematodes in soils is absolutely gigantic, and we seem to forget about it. They matter to us microbiologists because they eat bacteria usually.
Right.
I believe the number is something like 1 million nematodes per square meter of soil. Is that right?
That
Something
sounds about right.
Mm-hmm.
Huge number, and they matter to microbiologists. I think we got to pay attention to the guys who eat bacteria, not just the guys that get eaten by bacteria.
How many bacteria would there be in that same area?
There's 10 to the 4 species typically in an average gram of good fertile garden soil.
Wow.
There's something like 10 to the 7,
Wow.
10 to the 8, I think.
No,
gram.
10 to the 4,
Per
10,000 individual species. And then when you scale it up and ask the concentration effect, that's dependent upon all sorts of other variables.
But it tends to be high per
big.
gram.
It's
I mean,
big.
many more bacteria than nematodes, obviously.
Yeah, it's usually about 10 to the 11th, depending upon what methods you use to detect them per gram. So, yes, there are a lot more bacteria than nematodes, but the nematodes, they say, if you took away the rest of the soil and then looked at the Earth from space and just had the nematodes remaining, the maps of the world would look exactly the same, because most of the soil is inhabited by so many nematodes.
Wow.
Okay.
Wow.
But one of the interesting things Joe said is she introduced these small colonies. And if you think about Staph aureus as a pathogen, Staph aureus goes through something called the small colony variants. And I wonder if our staphylococcal scientist friends out there have looked for this sort of promoter element, because bacteria are great thieves. They know a good thing when they see it. And in this sort of regulation that we're seeing with this maternal adhesion switch, this mad operon, I wonder if that's what's going on with staphylococcal variants that become really small, because people have been trying to understand how these and the staph really makes these really tiny colonies, just like we saw with the M form of Photorhabdus. And so you wonder if we can begin to take something from our insect friends and ask to really get a handle on understanding how we can lock staphylococci in the good or maternal form as opposed to the Staph aureus that become the pathogenic form. And that may be a new area that people haven't thought about in terms of, trying to figure out what's going on with the pathogenesis of staph, especially in these multi-drug resistant staph that we're beginning to see sprout up everywhere.
Yeah, I think that's a good question.
I was thinking the same thing when I read it and wondering how many staph biologists were running back to their labs to look for mad switches in staph. Unless maybe they've already been discovered, but not that I know of. So I think it opens up a whole new mechanism for gene regulation and for the switching of bacteria from one state to the other.
So for either of these bacteria, the Photorhabdus or Theobacter, does anyone investigate the possible role of phages in these mutualisms, in these switches?
Well, I'm sure there's phage involved. The question is, are the phage encouraging the switching?
Yeah.
That's the question you're asking.
Yeah. Does anyone work on this? That's really what I want to know. And I don't know.
Let's assume they are.
Someone must be. If they aren't, they should be.
Important point, and it jumps at you in a way.
Yeah,
so.
yeah. Fantastic.
Great topic for another day.
It is.
Right.
I like this one. The last sentence. Chance promoter inversion appears to be an efficient mechanism in Photorhabdus to reversibly switch between conflict and cooperation with two hosts.
Well, the two papers really go together in that way, don't they?
Yep.
Yeah.
Beautiful.
I must say, I get more and more excited every time I run into another symbiosis, because there's something about symbiosis which tickle the mind. Really.
So you don't mind having a lot of symbioses, do you?
Can I try on you a silly thing? I wrote the power law of symbiosis. Can I read it to you?
Of course.
Sure.
Okay. Interacting genomes are more interesting than single ones by the nth power of the numbers, where n is a matter of personal preference.
It's great. Very good. Very good.
Does it work for you?
Yeah, it's good.
Yeah. You're such a romantic. You like symbiosis because you believe in love.
It also forces us to do math again, which is always good. You have to solve your equation.
But, Joe, it's just like humans.
My n number is very high.
I know.
It's just like humans, Joe. It's love, but it can also turn to death.
True.
Or hate or killing or whatever you want to say. It's terrible. I know. I'm throwing water on it, but it's the way it is. That's life. All right. Thanks for that, Joe. We have three emails, which I'd like to read, and all of you will be amused and entertained. The first one is from Pat, who writes, I'm a regular TWiM listener and assistant professor. I'm not going to say where because this will give it away. I was heavily involved in much of the data analysis for the Human Microbiome Project. As I was unable to attend the ASM General Meeting, I cringed when you and your co-hosts described the Human Microbiome talks at ASM as being glorified data dumps.
I don't think we.
Wait a minute.
I don't think we
that?
pray.
I think Michael said that.
I don't.
TWiM number
I'll
36.
Go back and listen.
This is most unfortunate. I was wondering whether you might be willing to have a guest, say me, to help get the general microbiologist through the weeds. As a former postdoc in Joe Handelsman's lab.
I know who it is.
I didn't want to say where she was. I can tell you that I'm very interested in making computational concepts more engaging and feel that we have an important job as scientists to view our research mission as part of our teaching mission. Anyway, if you are ever interested, please know that you have a standing offer from a microbial ecologist, bioinformaticist to help your listeners get excited about this amazing topic?
Well, for equal time for me, since they're blaming me for this topic, I was actually advocating for that, and I got an email from one of our listeners yesterday, and I wrote back. He also commented on the microbiome, and this is what I wrote back to him. I said that this meeting was a watershed meeting. The initial results on the microbiome were laid before the members of our society, and the challenge of discovery was both implicitly and explicitly offered. Results from this initial global study will be as transformational to our discipline as was the introduction of agar for the cultivation of bacteria.
Hey.
Thinking about how the community of microbes reacts with humans and in human health and disease processes will not only transform microbiology, but will similarly transform drug discovery, immunology, and medical diagnostics. Advances in each discipline when considering the microbiome may potentially lead to the development of new prevention strategies, treatments, or even potential cures for many of the chronic diseases currently plaguing humans. So that was what my take home from just sitting being overwhelmed by the volume of data. It wasn't a data dump. It was just the sheer enormity of what was being released at the meeting. I think that's what my takeaway from that was. And I apologize if I, disparaged any of the great presenters by saying it was a data dump. It was just, really remarkable is what that was my takeaway from it.
It was really a way of lauding people like this Pat person who does bioinformatics, because I think what amazed all of us was this, like you say, this massive amount of data and what it took to make sense of the data. And it's only because of the bioinformaticists that we actually can make sense of those big data sets. If there's anything we need as much or more than natural products chemists in microbiology, it's more bioinformaticists, especially more like this Pat guy who is very adept at not only the bioinformatics, but communicating it to biologists. So I think we should have him on our show.
Sure.
And in fact, along those lines, I started beating the bushes when I was at the general meeting, looking for people who could come on TWiM and help us begin to expand on this concept, because I'm certainly not an expert at being able to articulate the wonder that really needs to be displayed to members of our society to really get them excited about the materials that have come out.
So Pat is a assistant professor, University of Michigan Department of Microbiology and Immunology, and he writes, I enjoyed your visit last semester. That's me. I gave a seminar there, so must have met him. So Pat, we'll have you on. We'll get in touch, and you can talk about it. It's very interesting. On TWiV, we have the same issue when we do a paper that involves bioinformatic analyses. We just say we don't understand these and move on. And we've had listeners write in and say, get a bioinformaticist on to explain this. So it's the same problem. The next one is from Mark Martin, who all of you may know. And he writes, hello, Vincent, Michael, Elio, and the other contributors. I want to thank you so much for these podcasts. I've been having to sit around in traction for a pinched nerve a couple of times a week, and listening to TWiM enriches my mind while I feel like a science fiction character. Better than watching television in any case. I'm not sure that's such praise, right?
That's a pretty low threshold.
I really appreciate the interactions and information that are presented in all of your podcasts, but the comments on the ASM general meeting, which I had attended too in TWiM 36, resonated. The new card format for the posters was indeed wonderful for forgetful people like myself, and I have to agree strongly with Vincent's comment and Elio's quite gentle criticism of systems biology. Things are getting so complicated that few people can be generalists. I used to worry about eukaryocentricity in biology, but clearly I need to start worrying about colycentricity. I too thought that third slide at the opening session was a backgrounder for Salmonella phase variation, but decided I must not be getting it since the speaker was so clearly brilliant and engaged. So a solid background becomes vital, even for MacArthur Genius Grant winners. As our field sprints, it no longer marches forward. One final thing. I will be trying to introduce my undergraduates to TWiM in my fall microbiology course. The podcasts give an engaging and current feel for microbiology. I was going to say the ferment of microbiology, but I restrained myself. While my students might not want to listen for an hour, we do discuss journal articles, and I intend to assign some of the ones you have been covering, with the relevant TWiM assigned as background listening. Extra credit points often focus attention like a laser, I find. Best wishes to all, and thanks for what you do. Hope to see some discussion of microbial endocrinology at some point.
Oh, that's interesting.
But we had a couple of papers early on.
Yeah, we did, didn't we?
Yeah.
So is that the endocrine system of bacteria, or how bacteria influence the endocrine system of higher organisms?
Well, there are two ways to look at it, it turns out. One is that bacteria actually produce some compounds which resemble neurotransmitters and others, and have a direct effect. The other is that bacteria, seemingly, according to Mark Light, actually enjoy eating or living off things like norepinephrine, which is something you can only show if you grow them in a more natural sounding medium than LB or something like that. In other words, you put them in serum, which they don't like because there's not enough iron, but also other reasons. And if you add norepinephrine, the bacteria E. coli grows very well. So there may be a direct sensing of, behavioral chemicals by bacteria.
Well, and there's some new data that suggests that bacteria can influence depression in both humans and mice, and a lot more has to be done on that. But there are two very similar studies, one on each host, and it's quite remarkable the effect of the probiotics that they fed people and mice on depression. Well, the first thing you might ask is, how do you detect depression in mice? That's an interesting story in itself. But what I found out when I was reading that is that half of the serotonin receptors, so that's the depression-related hormone, the happy hormone as they call it, half of the receptors for serotonin are in the GI tract. So that's an answer in itself. Who else could be interacting with those receptors besides the bacteria?
Exactly.
P.S. The human microbiome descriptions are indeed hard to follow. I half expect to see advertisements for EnteroHarmony, the dating service that matches people based on their enterotypes. What about antibiotics then? Best wishes.
The antibiotic story is big because, that's one of the limitations is that everyone always puts up with their studies is that, the population that they got the samples from, they're very careful in telling you whether or not these folks have been on antibiotics and how long they've been not exposed to antibiotics. And that's often an exclusion criteria for some of the samples that were taken. And many of the people at the meeting who were presenting data made certain that they talked about whether or not their patient cohort was indeed exposed to antibiotics. So you bring up a really great point in thinking about the data that you're looking at in those studies.
And by the way, Mark, you might find that your students will listen for an hour because I assign my virology course to listen to TWiV, and they seem to enjoy it. So just let them loose. They might be okay. And our last email is from Spiros, who writes, I heard you read my email on the last TWiM, and I was super excited, really excited. And it looks like from the comments on the panel, it's something you guys would like to pursue. Please let me know when or if you guys get the time. So Spiros wrote about coming on and talking about control measures taken in the pharmaceutical industry. I am up to the ICAC podcast, but I have listened ahead to the most recent ones, obviously. The podcast talks about probiotics and the benefits, and I'm truly interested in them. However, I'd like to point out a recent MedWatch release, and here is the link. And he sends a link to an FDA recall. Sedona Labs issues a volunteer recall for iFlora Kids Multiprobiotic Dietary Supplement. due to possible health risk. This is a galacto-oligosaccharide product that is possibly contaminated with salmonella. So the company is recalling it. This isn't the kind of pharmaceutical I manufacture, but it's interesting nonetheless, and this is an example of what happens when things go very wrong in my industry. A lot of folks can get hurt. Each unit is going into a person, and I treat each one like a person. I also hope you guys mentioned what's going on in Cambodia. I saw 65 illnesses on CNN. Yeah. No cause for it right now. Of course, that may have changed. I've been out of touch for a while. But the news can't do as good a job as you guys can discussing this latest issue. Thanks again. Look forward to hearing future podcasts and hearing from you guys. So we're going to talk about the Cambodian outbreak tomorrow on TWiM, Spiros. So
it's got dengue involved.
So originally they had isolated enterovirus 71 from a number of these children, and now they're saying WHO says that it's a combination of enterovirus 71, dengue, and strep suis.
Oh,
does that make sense?
Well, it's a mutualistic infection, it sounds like.
But the real damage is done when the doctors in desperation administered steroids.
That's right. That's correct.
Yeah. That's what did it, because when people have a bad infection, that's about the last thing you can do, and there's not many choices. So they tried, and they made it much worse.
Yeah, the steroid treatment turned out to be a problem. So I don't know. We'll have to wait to see for the data. But Entro 71 on its own can be fatal as well. And they've had quite a few deaths so far this year in China from that infection.
Really?
So if you would like to send a question to TWiM, we're happy to get it. Send it to twim@twiv.tv. And you can find TWiM, of course, on iTunes or at the Zune Marketplace or microbeworld.org/TWiM. If you do like TWiM, go over to iTunes and leave a comment in the podcast area there. It helps us to stay visible so that more people can find us. And I'd like to wrap up TWiM 37 by thanking everyone. Joe Handelsman is at Yale University. Thanks so much for joining us today, Joe.
Thanks for having me. This was a great one. We had fun.
Always a pleasure, and look forward to the next couple of episodes with you.
Me too.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Indeed. Well, my pleasure. As always, but this was good.
Have you considered small things as we were talking here today?
That's all I ever do.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Well, thanks you. It's, again, another learning experience.
For everyone, especially me. Michael, what's the weather like down there? Is it really hot?
No, it's actually, well, since we're on the coast, it never gets really hot. Yesterday we had blessed rain, which we desperately needed, and today it looks like we're going to get the standard afternoon thunderstorm. So it's about 90, 95.
I don't know. That's pretty hot for me.
But it's a wet heat.
It's a wet heat.
Okay. It's a wet heat.
I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, Communications Director Barbara Hyde, and Chris Kondayan and Ray Ortega for technical help. And Ray's the guy who edits TWiM, so he takes out all the glitches that you never hear. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology. All right, great job. Very nice.
This was good, wasn't it?
Very good.
Yeah, this was really fun.
Good papers are always helpful, right?
Yeah, and once they fit together, it's always fun when I don't know if you planned it this way, but they really dovetailed beautifully.
You know, I didn't plan it. I just saw Elio had suggested the first one, and then I saw the second one. I think I saw on Microworld, and I said, "Oh, this is very cool." And it turned out that they work perfectly. So I'm happy for that. So hopefully next time can be just as good.
We should do something on the dengue story because LPS inhibits dengue infection. So and that's old, and so there's some weird connection between bacteria and dengue to begin with, and now
that's interesting.
Streptococcus thing makes it even more complicated.
So, do you know that polio requires LPS in order to infect the gut.
No, really?
So a paper came out in Science within the past year, which shows that polio, if you use notobiotic mice and infect them with polio, they do not get infected. And the reason is the virus binds LPS, and it's actually stabilized by binding to LPS.
Oh, my God.
Oh, maybe we should.
Is that on TWiM? Is that on TWiM?
Yeah, that's it. We did a TWiM. There were actually two papers in Science, one on that story, and then another one, mouse mammary tumor virus, which also infects mice through the gut, was shown to require also a bacteria in the gut. But in that case, the bacteria suppresses an immune response which would otherwise clear the virus from the gut. So when you take that bacteria out, you get this immune response, and the virus is cleared rapidly. So the viruses and bacteria are collaborating too.
Yeah, sick.
Let me ask you, how do you feel about piggybacking or recycling stuff from TWiM?
I don't have a problem because I think there's slightly different audiences, and you never can hear something too many times. So we could do those again in TWiM because your perspectives on the bacterial side would be interesting to get because we give the viral side. So.
Or we could do specifically interactions between viruses and bacteria because we have the polio story and the dengue story, and then I think there's an AIDS story of gut bacteria affecting HIV replication or something.
Yeah, that's getting really big now. I think the NIH is looking at that vis-a-vis the vaccine studies that they're going to do. That they're looking at the gut flora, and there also may be some bias, and that's why HIV selectively is able to infect women better than men from the gut.
Oh, really?
Yeah, I was talking to my chairman today, and we just recruited a new HIV virologist from Case Western, and that's what she's interested in. And he says I should really chat with her about these things, and so there may be something to it. I got to go and dig some stuff out about HIV in the gut, but he said that there's sex bias, which I hadn't heard about HIV.
Me neither. That's neat.
Well, I'll look into it. Maybe we can put maybe the next session we can do something on this because it would be cool.
That'd be great.
All right, thanks.
And the other thing I wanted to pass on is I just opened my issue of Microbe, and I just discovered that Bill Costerton passed away.
Yeah.
Really?
Yeah. I was like shocked from pancreatic cancer.
Several people died.
Yeah, Howard
this
Guest.
issue.
Yeah, Howard Guest passed away.
Yeah.
Howard Guest, who was not so young, but Costerton wasn't that old, was he?
No, 1934. So
late 70s. Wow.
yeah.
Huh. That's awful.
Well, unfortunately, on that note.
Unfortunately, but you all have a good weekend.
Yes, you too.
And look forward to the next one.
Yep, see you in 2 weeks. Thanks.
Yep. Bye-bye.
Great to talk to you. Bye-bye.